Hi all, welcome to episode 3 of Doorstep Science. Today we're joined by Dr. Olivia Reynolds, who is going to talk to us about the importance of habitat manipulation for promoting beneficial insects.
Hello everybody, my name is Olivia Reynolds and in this episode of Doorstep Science I'm going to talk to you about habitat manipulation in agroecosystems. Firstly, I'm going to give a general background and provide an example of where this is being used successfully at a large scale internationally. I will then introduce an Australian-wide project that is looking at habitat manipulation in Australian vegetable fields. In this episode, I will cover the findings of our field surveys from this project to date, and then in a follow-up episode, I will cover the results of our field trials and conclusions from this research. So let's get into this. Habitat manipulation is a form of conservation biological control. It involves the manipulation or ecological engineering of the habitat to enhance natural enemy, i.e. predators, parasitoids and pathogens, performance and their ability to control pests by improving the availability of resources. The aim of this strategy is to conserve natural enemies by providing flora that provides all or any combination of what we call SNAP, i.e. S, shelter, N, nectar, A, alternate prey, and P, pollen, SNAP. This practice favours natural enemies in agroecosystems which are often unsuitable due to high levels of disturbance. Indeed, one of the greatest obstacles in implementing habitat manipulation is the tendency, particularly in Western agricultural systems, for the application of heavy rates of pesticides. Although habitat manipulation may provide refugia for natural enemies from pesticide-treated areas, toxic residues left within the crop may repel or indeed disrupt subsequent activity. Habitat manipulation can involve a range of strategies such as trap crops, push-pull plants and flowering strips, just to name a few, and can be adapted for different agroecosystem requirements. An important consideration of applying these strategies on farm is that they are practical for farmers to implement and of course maintain. It is key that flowering plants selected for habitat manipulation to support natural enemies in agroecosystems display several important characteristics. Beneficial insects typically prefer white flowers, as shown in the image at the top right of your screen, a hoverfly feeding on a white rose flower. Although some of our pollinators, including honeybees and Australian native bees, such as the stingless sugarbag bee, have an innate preference for both white and blue flowers. Despite these preferences, this does not negate that natural enemies may be attracted to other coloured flowers, such as the lady beetle shown here feeding upon a purple flower at the lower right-hand corner of your screen. Natural enemies also tend to prefer open-face flowers where they can readily obtain nectar. Selectivity of plants is important as it is clearly not desirable to include plants that also promote pest species, and therefore careful consideration needs to be given to the crop type and the associated plant or plants chosen to promote natural enemies in a habitat manipulation strategy. For example, studies have shown that sesame flowers do not promote longevity or fecundity of the rice leaf roller, yet is known to support natural enemies that may attack this pest. There are several commonly utilised plants in habitat management scenarios, and these include alyssum, coriander, buckwheat, cornflower and sesame. However, each must be carefully considered and researched for each scenario that it is used. Natural enemies associated with plants come in many forms and include insects and arachnids such as hoverflies, lacewings and spiders. While the choice of plant species for habitat manipulation has largely centred on plants with suitable floral qualities to support natural enemies, there are numerous other benefits that can be attained through encouraging and supporting beneficials in agroecosystems. These include increased biodiversity, both safe and effective arthropod pest management, pollination services, not just from our bees, being those native or introduced, but also from the numerous other insects out there that visit flowers. Reduced pest pressure, reduced insecticide use, and increased yield may also be observed in several circumstances, as well as increased profitability. And of course, the aesthetic value of introducing these flowering plants to our landscapes. There are numerous successful examples and decades of scientific study to support the benefits of habitat manipulation in agroecosystems. A more recent study involved a large-scale habitat manipulation project in rice agroecosystems that was conducted across 16 sites in 3 countries, including China, Thailand, and Vietnam over a 4-year period. The bunds in rice fields were identified as ideal sites to plant suitable habitat, in this case nectar-producing plants, to encourage beneficial insects that would predate or parasitise key pests of rice. The farmers at each site were asked to only use pesticides if required, as opposed to following more traditional forms of spraying such as calendar or insurance-type spraying. The results revealed that this relatively straightforward and inexpensive intervention significantly reduced populations of two key pests, the rice-brown planthopper and the whiteback planthopper. Reduced applications of insecticide by 70%, grain yields were up by 5%, and this delivered an overall economic advantage of 7.5%. Moving now to a current study centred on Australia that has been funded through Hort Innovation, the focus of this study is to develop evidence-based habitat management guidelines for vegetable crops in 5 major vegetable production districts across Australia. The first component of the study has focused on identifying landscape characteristics that promote beneficial insects. This has involved a survey targeting 6 focal crops, including carrots, brassicas, beans, sweet corn, lettuce and capsicum across every Australian state. The second component is looking at the use of flowering strips in brassicas and sweet corn and trap crops in brassicas. These trials are currently underway and we won't discuss these further here today, but the results will be discussed in a follow-on episode of Doorstep Science. As part of the first component of our vegetable project, we surveyed nearly 500 vegetable fields across the 6 states of Australia, including those managed conventionally and organically. This was performed over a 1-year period and aimed to sample each focal crop type in each season the focal crop was grown in each state. In each case, we sampled a 10-metre strip from each edge of each focal crop that was bordered by a different land use or vegetation type. If a focal crop was bordered by the same adjacent land use more than once, only one edge was selected to sample. The diagram here shows an example of what a sampling regime might look like for a particular focal crop. In this example, as no 2 edges had the same adjacent land use, every edge was sampled, with 2 edges sampled in 2 different sections because those edges bordered multiple adjacent land uses. Adjacent land use was highly variable and could include any combination of crop, shelter belt, roads, dams, riparian vegetation, and weeding flowering plants, just to name a few. Our vegetable surveys provided us with some great insights into on-farm landscape characteristics that promote beneficial insects. Here I present some of our early findings. It was evident that adjacent land use types impacted the biodiversity and density of pests and natural enemies in our focal crops. Our results also showed that encouraging and supporting non-crop vegetation, particularly shelter belts and riparian strips, promoted natural enemies in nearby focal vegetable crops. It appears to be advantageous to arthropod community composition to separate vegetable crops with other land uses crop types. Such strategies can assist in minimising the build-up of a pest species in a particular crop type, for example, particularly where there are a succession of growth stages of the same crop type, host permitting the pest to readily move between suitable stages. Interestingly, we found that pest numbers were quite similar between vegetable crops that used biological pesticides and those that were conventionally managed, i.e. used synthetic pesticides. However, natural enemies were significantly higher in crops where only biological insecticides were used. This suggests that natural enemies, in conjunction with biological-based pesticides, can adequately check pest outbreaks when present in sufficiently large numbers by virtue of the presence of suitable non-crop vegetation. Collectively, the findings from our vegetable field surveys, together with the results of our in-field trials testing the efficacy of flowering strips and trap crops, will be used to inform the development of our habitat manipulation management guidelines for Australian vegetable growers.
Thanks for joining us today. We'll see you next time when Dr. Olivia Reynolds will discuss the role that native parasitoid wasps play in controlling biosecurity threats. Doorstep Science. It's our way of delivering good science straight to your door.